Hi, my name is Kraus, and I'll be discussing an examination of antibiotic resistance genes in Pseudomonas chlororaphis in response to bacteriophage therapy. One of the rising problems in healthcare currently is the increase in antibiotic resistance in bacteria, leading to the emergence of multi-drug resistant bacteria. This is an issue because not only are bacteria becoming more resistant to antibiotics used in treatment, but the rate of antibiotic development has also slowed dramatically over the past decades. So, in the combination with the decrease in new effective antibiotics, coupled with the rise in the frequency of bacteria resistant to these antibiotics, it is becoming a very troublesome issue that must be addressed in the near future. Additionally, the development of these new antibiotics proves very costly and time-consuming, therefore adding an additional burden onto this issue. As you can see down here, initially a bacteria may have resistance to a certain antibiotic, but over time the bacteria may acquire resistance through various mechanisms that lead it to become more resistant to antibiotics. In order to understand where this problem comes from and how to progress from here, an understanding of different antibiotic resistance mechanisms in bacteria is warranted. So, antibiotic resistant bacteria are a problem, but how did they arise in the first place? Well, two main mechanisms exist for this: intrinsic resistance and acquired resistance. Intrinsic resistance utilizes genes already inside the cell in order to confer antibiotic resistance. And examples include efflux pumps, outer membrane proteins, and electron transport chain components. An example can be seen here with efflux pumps and outer membrane proteins. An antibiotic can enter the cell, therefore targeting a cellular process, eventually leading to the cell's death. However, efflux pumps coupled with outer membrane proteins can grab a hold of this antibiotic before it does any damage, export it out of the cell, therefore leaving the bacteria resistant to that antibiotic. Acquired resistance also propagates antibiotic resistance genes. However, the genes aren't inside the cell and are acquired from another place initially. Strategies for this include transformation, conjugation, and transduction, which will not be discussed here. However, the general principle for acquired resistance is the transfer of a resistance gene from an antibiotic-resistant bacteria or the environment into a non-resistant bacteria, and now that bacteria has the resistance gene and is resistant to that antibiotic. Bacterial strains have a unique combination of both intrinsic resistance and acquired resistance, making each strain unique and increasingly difficult to treat. Another method to combat these multi-drug resistant bacteria includes the use of viruses. Viruses that only infect bacteria are called bacteriophages. Since they do not attach to or enter human cells, they do not harm humans or other animals that they are not specific for. However, these viruses will infect bacterial cells, eventually leading to their death. The bacteriophage begins by attaching to the surface of the bacteria, where it then injects its genetic material into the bacteria. The viral genome then uses a bunch of host cell machinery such as ribosomes and other proteins to produce a bunch of smaller viruses inside of the bacteria. Eventually, this bacteria will lyse or split open, releasing these new viruses into the environment to infect other bacteria. A potential problem exists due to the bacteria eventually evolving resistance to a virus, therefore not allowing the virus to enter, reproduce, and lyse the bacteria. While this may seem like a disadvantage, the evolution of bacteriophage resistance does pose potential advantages. One of these advantages has been shown that bacteriophage-resistant bacteria often have altered or increased antibiotic susceptibility, allowing for potential new treatments involving both viruses and antibiotics working together for these pesky multi-drug resistant bacteria. So, a potential solution includes combining phages and antibiotics for treatment of these multi-drug resistant bacteria. This is also known as dual therapy. It involves the simultaneous use of phages and antibiotics to accomplish effects that the singular use of a phage or the singular use of an antibiotic cannot accomplish alone. Initially, the phages will kill most bacteria, and the surviving population may have arisen from a mutation that renders it resistant to the virus, but also more susceptible to antibiotics. Afterwards, the administration of antibiotics to clear the remaining bacteria the phage did not kill will eliminate the infection from the patient. So as you can see here, through a quick illustration, there's an initial population of bacteria, and upon exposure to this phage, most of the bacteria die, but a small amount of the phage-resistant population remains. Then the administration of antibiotics will kill the remaining bacteria, leading to no more bacteria and no more infection. Now, the brief overview of viruses, antibiotics, bacteria, etc. will allow for greater understanding of the specific aims and hypothesis of the study. The main goal of the research is to understand how the evolution of bacteriophage resistance in Pseudomonas chlororaphis affects its susceptibility to antibiotics, providing insight into new areas of treatment regarding antibiotic-resistant bacteria. The hypothesis is that bacteriophage-resistant mutants will show increased susceptibility to certain antibiotics, providing a list of phage-antibiotic combinations that display synergistic activity. The experiment is divided into three main phases, with the first phase being the antibiotic susceptibility testing of the 14b11 parent strain. Eventually, this parent strain will be used to create mutants that are resistant to bacteriophage infection, and antibiotic testing of the mutants will follow. The last phase involves sequencing and comparison of candidate genes in the parent strain and the mutants to determine if any of these mutations could affect susceptibility that has been seen. The antibiotic susceptibility testing of the parent strain can be seen here. 10 total antibiotics were tested, where the solid lines in the graph represent the control trials, where the bacteria was grown without antibiotic, and the dashed lines represent experimental trials, where it was grown with a certain concentration of that antibiotic. Some of the key results indicate that the parent strain is resistant to some antibiotics, while highly susceptible to others. If it was resistant, that means that the control and experimental growth rates were similar, as seen with ampicillin and sulfamethoxazole on the right-hand side of the graph. If it was susceptible, then there's a large difference between the control and experimental bars, which can be seen with rifampicin and gentamicin in the center part of the graph. Another interesting thing to note is that resistance varied within specific antibiotic classes. For example, the beta-lactam group includes meropenem and ampicillin in the study. Both target the same process within the cell, therefore, one would expect that the resistance or susceptibility levels would be the same. However, this is not the case. The parent strain was very susceptible to meropenem, however, it was not susceptible at all to ampicillin. The same thing can be seen with the aminoglycoside antibiotic class, which just targets a different process in the cell. Phase 2 involved exposing the 14b11 parent strain to the PC1 bacteriophage in order to create bacteriophage-resistant mutants. An overview of the procedure used can be seen here in this figure. Basically, the 14b11 parent strain was combined with phage lysate, which contains a purified solution of the virus, and allowed to incubate for 48 hours total. Then, a small portion of that was taken, diluted across multiple factors, and then plated and allowed to grow overnight. The results of these dilution plates can be seen here, with 1 being the most concentrated solution and 6 being the least concentrated solution. As you can see, the bacterial growth varies from very dense to just isolated colonies. The growth of these isolated colonies of bacteria infers that this bacteria can grow in the presence of the bacteriophage, making it a potential phage-resistant mutant. So as you can see, the prolific growth indicated many mutants across many plates. Therefore, to focus more, only 27 potential bacteriophage-resistant mutants were selected for a further study. Phase 2 involved taking the mutants that were created and then testing them against all the antibiotics that the parent strain was tested against. Overall, 27 mutants were generated and tested against 10 antibiotics total, creating a lot of different results, which can be summarized in the color graph seen here. Basically, the green indicates that antibiotic and mutant combination grew better than the parent strain, therefore it was less susceptible to the antibiotic. The images in the red and purple indicate that the mutant grew less in the antibiotic compared to the parent strain, therefore it was more susceptible than the parent strain. The ones in gray indicate ambiguous results and will not be discussed at this time. Some of the key results are that 26 of the 27 mutants showed increased susceptibility to at least one antibiotic tested. Certain specific mutant antibiotic combinations also displayed very large changes, and these were selected for further study. These were broken down into antibiotic class, which was inferred from the previous graph. The ones in the black boxes indicate, through a second round of antibiotic testing, that these did in fact grow less compared to the parent strain when exposed to the same antibiotic. And again, the second round of antibiotic testing followed the same procedure as the parent strain in the first graph of the study. So, candidate gene examination. Why these genes? Overall, 8 genes were selected for study: MEX-E, MEX-F, OpRN, MEX-G, MEX-H, MEX-I, OpMD, and SIO-E. The first 3 of these genes combine to form the MEX-EF OpRN efflux pump, which exports certain antibiotics from the cell, as was shown in the antibiotic resistance slides earlier. The next 4 genes combine to form the MEX-GHI OpMD efflux pump, which serves the same function just with different substrates. Lastly, the SIO-E gene encodes a cofactor in the electron transport chain, which is involved in creating the electrochemical gradient across a cell's surface. By altering this gene, it can alter the electrochemical gradient, which affects the ability of some antibiotics to move in and out of the cell. So, as you can see here, the MEX-EF pump confers resistance to tetracycline, ciprofloxacin, and chloramphenicol, where the MEX-GHI OpMD pump only confers resistance to ciprofloxacin. The SIO-E gene confers resistance to aminoglycosides, as the 3 are listed here. Additional antibiotics were tested just to see their effects on the mutants and the parent strain. In order to compare these candidate genes between the mutants and the parent strains, PCR and gel electrophoresis were performed on the candidate genes. PCR is basically a procedure that allows you to take a specific section of DNA and amplify it through a repetitive process, giving you a product at the end. Gel electrophoresis allows you to separate these products from PCR based on the size of the fragment that you choose to amplify. It also separates other unwanted products during the process. Some of the key results of the PCR and gel electrophoresis procedures can be seen here. As you can see, some primers produced clear amplicons for the target gene, as seen by the SIO-E gel right here, where amplicons are seen pretty consistently across all mutants. However, others did not produce clear results or any amplicons at all, as seen by the gel here. This was a very tedious and repetitive process and involved lots of modifications to the procedures. Eventually, to focus on more promising genes and specific mutants did yield amplicons, as seen by the figure here, where OpRN and SIO-E were increasingly focused on due to their success. However, these amplicons were not at the right DNA concentration to be isolated and sent for sequencing. Eventually, sequencing results of SIO-E were determined to be inconclusive, therefore complicating the end results of the study. So, to conclude, the parent strain displayed variable resistance to a wide range of antibiotics. The mutants that were generated from the parent strain were also easily identified, and many did show increased susceptibility to at least one antibiotic. In order to save time and effort, a select few mutants that displayed increased susceptibility were further studied and isolated through the PCR and gel electrophoresis procedure. However, the candidate genes involved in antibiotic resistance were unable to be sent for sequencing, but the presence of amplicons in this entire process shows that they are inside the cell, but do require further modifications to the procedures in order for proper extraction and sequencing. Future work includes improving the PCR and gel electrophoresis procedures of the candidate genes, either by improved primer design, PCR methods, or adjusting of annealing temperatures. Also, examining additional efflux pumps in the parent strain and mutants would prove interesting. Aside from the MEX-EF and MEX-GHI efflux pumps, a MEX-AB efflux pump also exists, which confers resistance to different antibiotics. Therefore, studying it in the future would prove beneficial. Lastly, study of successful synergistic combinations in vivo or in an organism would prove beneficial. By taking an infected rat and administering phages and antibiotics at the same time, the measurement of the remaining bacteria can prove this dual therapy's effectiveness compared to antibiotics alone or viruses alone. I would like to thank Dr. Strand for her continued support and assistance throughout the senior IS process, as well as individuals in the Strand IS Lab Group and the BCMB program for making this all possible. I'd also like to thank the Copeland Fund for financial assistance, as well as the Wooster football program and my family, friends, and teammates for providing endless support along the way. Thank you.